Hello everyone, I am Sneha, a third year MBBS student at AIIMS Nagpur, and today we'll be doing previous year questions from the chapter Reproductive Health. And one to two questions are being asked from this particular chapter repetitively every single year. So we need to make sure that we are doing all the previous year questions properly. So we'll start with the first question. We have question number one, which is in which of the following techniques the embryos are transferred to assist those females who cannot conceive. So this is an example of assisted reproductive techniques. Okay. And they have asked us to name the technique in which embryos are transferred. So in ARTs, we can have transfer of either sperms, either ovum, either embryo. Okay. So in this particular one, they have asked us about the transfer of embryo. So firstly, we'll just take a look at all the options. We have GIFT, we have ZIFT, we have ICSI, we have IUT. So GIFT is what? It is gamete intra fallopian transfer. Then we have T, which is zygote intra fallopian transfer. Then we have ICSI, which is intracytoplasmic sperm insemination. We have intracytoplasmic sperm insemination. And then we have IUT, which is intrauterine transfer. Okay. So let us have a quick look at all these techniques. In GIFT, there is a transfer of gamete. It can be either sperm or it can be either ovum. In zygote, that is ZIFT, we take, for example, you might have heard about test tube baby. So it is like that. In an external in vitro, we are uh, doing the process of fertilization where we are placing the ovum and sperm in contact. Fertilization is taking place, and then the zygote, it is transferred into the fallopian tube of the female who wants to conceive. In ICSI, we take an ovum and sperm is inseminated into it, and IUT is intrauterine transfer. So, ART, in which embryos are transferred. So, embryos are transferred either in ZI, which is zygote intrafallopian transfer, or IUT, which is intrauterine transfer. So, in this case, also embryo is transferred. Okay. And this was all. So in all of these, in GIFT, gametes are transferred. And in ICSI also, gametes are transferred. The sperms are artificially inseminated. Moving on to the next one, which of the following sexually transmitted disease that does not specifically affect reproductive organs? So commonly, we have conditions like vaginitis or cervicitis. So, what are these conditions? Vaginitis, it is the inflammation of vagina in female. The inflammation of vagina in females, that is known as vaginitis, and inflammation of cervix, it is known as cervicitis. Okay. So, these are the like in these conditions, they are sexually transmitted diseases, and there is an reproductive organisms they are affected. There are some conditions, for example, chlamydiasis. We'll have a look at all of these. We have option A as chlamydiasis. So chlamydiasis, it occurs due to the bacterium chlamydia. And in this, we can see the deformation on abnormality in the reproductive organs. So this particular infection, it affects reproductive organs. And how does this affect? They can be discharged coming out. There can be formation of small, we can say ulcers can be formed or papules can be formed. So chlamydiasis, it affects the genital organs. Next, we have AIDS. Is AIDS a sexually transmitted disease? Yes, it is a sexually transmitted disease. And what is the virus involved? It is HIV. HIV, RNA virus. Okay. So HIV, it is a RNA virus, but what does it affect? AIDS is acquired immunodeficiency syndrome. What is the full form of AIDS? We have acquired immunodeficiency syndrome. Okay. So acquired immunodeficiency syndrome is the one. And what is deficit in these persons? There is a affected immune system. Okay. So immune system is affected in these people. Okay. So AIDS, in short, it doesn't affect reproductive organisms, reproductive organs. So reproductive organs, they are not affected in this particular disease. Next, we have genital warts. We have B as genital warts. So yes, genital warts, it affects reproductive organs. Next, we have so hepatitis B. It is transmitted through sexual intercourse or sexual contact. So it is transmitted through sexual contact. But what is affected in hepatitis B? It mainly affects liver. Okay, so liver is affected due to which cirrhosis of liver takes place, and then there is elevated liver enzymes. But this particular disease, it does not affect our reproductive organs. Next, we have syphilis. Syphilis is due to what syphilis is actually caused by our bacteria. Okay, so syphilis, it affects our reproductive organs. Okay, so reproductive organs are particularly affected in this condition. Herpes, we have uh, genital herpes. It is caused by what? It is caused by herpes simplex virus. So herpes simplex virus causes this particular disease and it affects reproductive organs. Till now, there have been just two of the infections that do not affect our reproductive organs, that is either hepatitis B or we have AIDS. So, option D, which consists of both of these options, is the right one. So, hepatitis B and AIDS, they are transmitted through sexual contact, but they do not affect our reproductive organs. Okay, moving on to the next one. Which of the following sexually transmitted disease is not completely curable? So we have question number 3. The disease which is not completely curable is genital herpes. Okay. So genital herpes is not completely curable. There can be a latent phase. Okay. So let us talk about genital herpes. It is a viral infection. Viral infection, and it is caused due to which virus? Herpes simplex virus. So herpes simplex virus causes genital herpes. Can be either HSV1 or it can be HSV2. Okay. So at present there is no cure for type 2 herpes simplex virus. And other non-curable STIs are what are in this. We'll just make a list of the non-curable sexually transmitted infections. So it is either herpes or we know AIDS is non-curable. And next we have hepatitis B, which is not curable. Okay. So we have vaccines available for hepatitis B, but we don't have any um, particular vaccines for AIDS. Okay. Moving on to the next one, we have a couple where the male is having a very sperm count. So the sperm count of the male is very low. So if the sperm count is low, what do we need to do? We need to do artificial insemination. Hmm. Okay. And in this process, what we do, we inject the sperm directly into the vagina or uterus. There is no in vitro fertilization taking place. Where is the semen injected? It is injected into vagina or uterus. Okay. So this right here is artificial insemination. We'll just have a look at the other options. We have gamete intracytoplasmic fallopian transfer, in which ovum can be transferred or sperms can be transferred. But as we know, there is already very low count of sperm, so this is of no use. ICSI is also not used here, and intrauterine transfer, it is a type of in vitro fertilization where the fertilization it takes place outside the body. So what is IVF? It is in vitro fertilization, and the process of fertilization it takes place outside of the body, right? Moving on to the next one, we have ectopic pregnancy. So question number 5 is ectopic pregnancy. Ectopic pregnancy is a type of pregnancy. We know that normally in a normal female, when she conceives, and if she is having a normal pregnancy, the zygote or the embryo is the one that gets implanted. Embryo, it gets implanted where? It gets implanted in the uterus. But in case the embryo, it has implanted some other place, at some other place, other than what? Other than the uterus. Then this phenomena right here is known as ectopic pregnancy. What are the sites where this ectopic pregnancy can be implanted? It most common is fallopian tube. Okay, so these type of pregnancies they are not commonly viable. So there are high chances that we would have to terminate this pregnancy. Okay, so in ectopic pregnancy, the implantation of embryo it doesn't occur in the uterus, but at some other sites. So it is also known as tubal pregnancy. And moving on to the next one, a childless couple can be assisted to have a child through a technique called GIFT. So GIFT is what? GIFT is the short form for gamete intrafallopian transfer. So in this process, the gametes they are transferred into the fallopian tube of the woman who wants to conceive, and as the fertilization, where does this fertilization takes place? This fertilization takes place inside the uterus itself, inside the female uterus or fallopian. And so it is an example of in vivo fertilization. Okay. So, where is it used? It is used for infertile women who are ovulating, but they have blocked fallopian tubes. So, we know if we draw a image of this right here, is the ovary. This is the fallopian tube, and it is blocked right here. There is a block. So, the woman, she is ovulating, she is releasing an ovum, she is ovulating properly, but due to this blockage, the egg is released here. What is this? It is the egg or ovum. It has to reach here. It has to reach here, but it is not able to reach here. So in this case, we'll take out the ovum and we'll artificially insert it into the fallopian tube. Okay. Next, we have assisted reproductive technology under IVF, in vitro fertilization. So question number 7 is in vitro fertilization. So what is in vitro fertilization? That the fertilization is taking place in vitro, that is outside the body. Outside the body, our fertilization is taking place. So what all is involved in this? Do we insert the ovum into the fallopian tube? In this case, the fertilization will take place in the body of the women itself. So A option is incorrect. Zygote into the fallopian tube. Zygote is usually not. In this case, the fertilization process is taking place outside the body only because inserted. They are inserting the zygote, but the zygote is not usually inserted. We insert with 16 blastomeres, and this is inserted into the fallopian tube. Okay. So, embryo with usually 32 blastomeres is actually done in some other techniques. So, in case of IVF, we have a process of manually combining an egg and a sperm in the laboratory dish that is outside the body, and this right here is an in vitro process. So, the process, it involves stimulating a ovulatory process. We remove the ova. The ova is removed. Ova is removed, and it is fertilized with the help of sperm in a fluid medium. So, fertilization takes place with the help of sperm. Where does this take place? In a fluid medium, in a laboratory. And then it is inserted into the fallopian tube of the women. Next, we have artificial insemination. So we have our next question, which is artificial insemination, question number 8. Artificial insemination is an example of in vivo fertilization. So, artificial insemination is an in vivo process where the sperms of healthy donor, they are inserted into the vagina. And where does the fertilization takes place? The fertilization, it takes place inside of the female body. Okay. So this process right here is in vivo. So option B is the correct one. Okay. Next one, we have the stage in which uh, the after the induced fertilization of ova in the laboratory, in which stage do we transfer the into the uterus? Question number 9. We have in intrauterine transfer, and this is a type of in vitro fertilization. Why is this a type of in vitro fertilization? Because the fertilization, it is placed in a laboratory outside the human body. So in vitro fertilization, we usually transfer the morula stage. And how many blastomeres are present in a morula? 8 blastomeres are present. Okay. 8 blastomers are present in the morula stage, and it is then transferred into the uterus. The 8 cell stage, it develops by which day? By day 3. Okay. So by third day, we morula is formed. How many cells do a morula have? It has 8 blastomers, and then it is transferred into the female body. The test tube baby program is a part of in vitro fertilization. So question number 10th, we have test tube baby program. In vitro fertilization. And what is involved in it? Intracytoplasmic sperm injection. It is not usually given. It is a part of in vivo fertilization. Intrauterine insemination, also the fertilization is taking place inside the female body itself. Also in gamete intrafallopian transfer, the fertilization is taking place inside the female body only. Hmm. So in zygote intra ZIFT, which is zygote intrafallopian transfer, is usually employed under the test tube baby program. Okay. So we have medical termination of pregnancy. Medical termination pregnancy. Medical termination of pregnancy, it is considered safe up to 12 weeks. So up to 12 weeks, it is considered to be safe. Total of 37, after 38 weeks, we consider it as pregnancy. So up to 12 weeks, MTP is considered to be safe. Okay. Next, we have a technique called as gamete intrafallopian transfer. So where is this gamete intrafallopian transfer recommended? So the this process, it usually takes place via insert the gamete inside the uterus or fallopian tube of the women. So it is the fertilization is taking place in vivo. We'll be having the fetus inside the uterus only, and the environment is also proper. But in the case that we have a woman, this is a woman who cannot produce an ovum, we'll have to take an ovum from some donor. So this donor will provide the ovum, and it will in the women who wants to conceive, and then fertilization will take place. Okay. So GIFT is recommended for females who cannot produce an ovum. Okay. So in this process, the eggs of the donor women are removed, and in the form of a mixture with sperm, they are transferred into the fallopian tube. The fallopian tube and other structures are normal. There will be proper fertilization, there will be plantation and development. Okay. So which of the uh, following correct statements are correct. Okay, so question number 13, we have to tell the statements which are correct. 1, 2, 3, and 4. So they have said that the MTP during first trimester is generally safe, which is correct one. So generally chances of conception are nil until mother breastfeeds the infant up to 2 years. This process is known as lactational amenorrhea. We have lactational amenorrhea, and it is not valid up to 2 years. It is around 3 to 6 months only. So this one is incorrect. This one is correct. Third option we have intrauterine devices like copper T are effective contraceptive. Yes, we have copper containing intrauterine devices which are toxic to the sperm, which is uh, usually considered to be safe. And next we have that contraceptive pills, they can be taken up to 7 days, that is 1 week. The longest you can take a pill is after 72 hours. We have a pill named Ulipristal, and this Ulipristal has the maximum capacity, so it has to be taken within 72 hours. 1 week, we do not usually wait for the contraceptive. So the correct one inside in this options is first and third option. So first and third options is the correct one, which is our option C. You need to make sure that you are reading the question well so that you know they have asked the correct one or the incorrect one. There is just a slight change. They would give which two of the following are incorrect. Answer would be second and fourth. So it would be a totally opposite question. Okay. Test tube baby is a case of in vitro fertilization, and it has usually undergone development in a test tube. So development doesn't take place in test tube. We need to make sure it is not that we are just the fertilization is taking place in test tube and the baby is developing, and it is not the case. Only the fertilization process is taking place, and after fertilization, we transfer it to the female body. Okay. So option C, which is that the test tube baby is born out of the technique of fertilization in vitro, is the correct one. Okay. Moving on to the next one, we have sexually transmitted diseases. Question number 15th, we have to tell about STDs. So gonorrhea, yes, it is a sexually transmitted. It is transmitted. The organism is Neisseria gonorrhoeae. Malaria is not sexually transmitted. It is transferred by the mosquito bite. Aedes, we have malaria. Okay. Then we have genital herpes. Yes, it is transferred by sexual contact. AIDS is transferred by sexual contact. Filariasis is not by sexual contact. Then we have syphilis, which is transferred by sexual contact. So syphilis is transferred by sexual contact. Correct answer is option D. We have gonorrhea, we have syphilis, and we have genital herpes. All of these three are transferred by sexual contact. Okay. Let us look at the causative organisms. We have Neisseria gonorrhoeae, which is a bacteria. Then we have herpes simplex virus, and for syphilis we have treponema. Okay. Moving on to the next one, we have tubectomy is a method of sterilization. Tubectomy, in which we cut the tube. What tube? Fallopian tube. So question number 16, tubectomy. Tomy means cutting, incision, and tube, fallopian tube. So in this, a small part of fallopian tube is either removed, it is either removed or it is either tied up. Okay. So this is the correct answer, which is option A. We have another such term which is known as vasectomy. So we'll just go by the name. Tommy means removal or incision, and vas deferens. So it takes place in, it is a method of contraception in males, and it is a terminal method, like it is irreversible method. So it is a method of contraception in males. Moving on to the next one. So tubectomy, they have shown the figure, which is also known as tubal sterilization. It is a process done on women as per reversible method. Irreversible or permanent method. Okay. Next, we have which of the following is an hormone releasing intrauterine device. So, multi-load 375 is a copper releasing one. So, we have question number 17. Multi-load 375. It is a copper releasing intrauterine device. Then we have LNG 20. It is levonorgestrel. And levonorgestrel is a type of progesterone. So this is an hormone releasing intrauterine device. Then we have cervical cap, and they are a method of barrier method of contraceptive. So we have cervical cap and wall. These are what? These are barrier methods. Okay. Moving on to the next one. So intrauterine device. It is LNG 20. Why is it called LNG 20? Because it releases 20 milligram of levonorgestrel in 4 hours. Okay. Moving on to the next one. Which of the following cannot be detected by detected in a developing fetus by amniocentesis? So amniocentesis is a process, invasive process, in which fluid is taken from amniotic fluid is taken for diagnostic process. And what do we diagnose? We can either diagnose sex of a fetus, and due to this very reason, it is now illegal. Then we can diagnose any chromosomal abnormality. What all chromosomal abnormality do we know of? It can be either Turner's, it can be Klinefelter's, or we can have Down syndrome. Okay. So these are the three options. We have A, B, and D. Jaundice is not usually diagnosed by amniocentesis. Okay. So one of the legal method of birth control is, what is the legal method? We can have abortion by taking an appropriate method. The first one we have, question number 9. Abstinence is a natural method. There is no such method mentioned as option B, which is by having coitus at the time of day. Then we have premature ejaculation. This is also not a method. And D is the legal method that we can undergo abortion up to what? Up to 12 weeks. Abortion is legal. After 12 weeks, it is, we need to take some doctor. We have a panel of a doctor that we need to take advice from. Okay. So we have option D. We have which of the following is not a method of contraception? Question number 20. We have tubectomy, which is a permanent method, permanent sterilization. Next, we have condoms, which are a barrier method. Next, we have pills, which is the of oxytocin and vasopressin. These are not actually any method of contraception. And D, we have lip slew. which is also a method of contraception. So oxytocin is a birth hormone. It causes contraction of muscles, and it is given during parturition. So it acts on the smooth muscles of our body, and it stimulates their contraction. Okay, and vasopressin has no role. So they have tied the fallopian tube in this. So question number 21. Fallopian tubes are tied. So this is what? This is tubectomy. Next, we have most widely accepted method of contraception in India is intrauterine devices. This is a logistic based question. We have IUDs, that is intrauterine devices, as the most accepted method. So we insert the IUD through the cervix. It can be either copper containing or hormone containing. Next, we have copper ions that are released from the copper containing intrauterine devices. And what is the function of this copper? It actually increases the phagocytosis. So, question number 23 we have copper containing IUDs. So, what is function of this copper? It actually suppresses sperm motility. Okay. Next, we have match the following. Question number 24. What do we have? The pill, condom, then we have vasectomy, and then we have copper T. So, copper T, it is a process in which there is uh, the copper ions that are present in a copper T. It is an intrauterine device, so it prevents implantation. Okay, vasectomy, we block the vas deferens. So the semen, it contains no sperm. Condom is a barrier, so it prevents sperms from reaching the cervix. Okay, and I pill, it prevents ovulation. It contains a combination of estrogen and progesterone, and it prevents ovulation. So the pill, it prevents ovulation, that is option B. Condom, it prevents sperm from reaching the cervix, which is option A. Vasectomy, in vasectomy we have semens but no sperms, which is option D. It prevents implantation, which is option D. So we have B, A, C, D, which is our option D. Okay, which is option A. Okay. So option the pill, it prevents ovulation. This right here is option C. Sorry, I did it my mistake. Condom, it prevents the cervix from reaching. It prevents the sperm from reaching cervix. Then we have vasectomy, in which there is a semen, but it doesn't contain any. So we have option E. Next, copper T is a device. What is copper T? Intrauterine device. It is sort of like this. Okay. So it is like this, and it releases copper ion. What is the function of these copper ions? These copper ions, they decrease the motility of sperm. Or other one is that they prevent implantation. So they'll decrease the motility of sperm so that fertilization doesn't take place. In case fertilization takes place, they'll not allow the implantation of embryo. So it acts as a contraceptive method. Moving on next, we have question number 26, which of the following is a correct statement? We have intrauterine devices. Does it suppress gametogenesis? No. Intrauterine uh, device, they have no effect on gametogenesis. Intrauterine devices, no effect on gametogenesis. Next, we have IUDs. They need to be replaced after. There is a shelf life for each IUD, after which they need to be replaced. And no, the user cannot just insert them by herself. We need proper medical intervention to insert it. And IUDs, it increases the phagocytosis of sperms in the uterus, which is the correct option. Okay. So moving on to the next, we have the permissible use of technique of amniocentesis. Amniocentesis was used to determine the sex of baby, after which it was termed as illegal. But we can still do amniocentesis in what cases? We can do it to detect any genetic abnormality. Okay. So it can be done legally. This one is legal. And for determination of sex of baby, it is illegal. Amniocentesis, what is the technique? In this technique, we can we take out the amniotic fluid. So amniotic fluid is taken out. And what do we do with that amniotic fluid? It is used for screening, either for any genetic abnormality or for sex of the fetus. We just screen. We cannot reverse the sex of the fetus, which is given as option C. Okay. And option B, which is that any chromosomal abnormality in the fetus can be detected, it is the correct option. So we have question number 29th, which is which of the following contraceptive method do involve a role of hormone? So hormone is used in pills. Okay. So pills, they use in them which hormones? Usually estrogen and progesterone. These hormones are included. Then we have lactational amenorrhea. What is lactational amenorrhea? Lactational amenorrhea just after childbirth. There is an increased level of prolactin due to which ovulation doesn't take place. Ovulation does not take place. Okay. And what does it result into? It results into contraceptive benefit. Next, we have emergency contraceptives. Emergency contraceptives also have estrogen and progesterone in them, which do not allow the implantation of the embryo. Barrier method actually uses doesn't use any hormone. There is a physical obstruction. So barrier method uses what? Physical obstruction. Okay. Copper T doesn't use any hormone. There are copper ions which increase the phagocytosis of sperm and they decrease the motility of sperm. So we have option A as the correct one. Okay. So prolactin is a major hormone which is responsible for milk production and it is present in sufficient quantities in almost all women and it then acts as a contraceptive benefit. We have to select the hormones that release the hormones the releasing intrauterine devices. So we have question number 30. Option A, we have warts. What are warts? They act as barrier method. Then we have LNG 22. It is a hormone releasing. Next, we have multi-load 375. Multiload 375, it releases copper. Progesterone, it is a hormone containing. So, two hormone containing amongst these are what? It is either LNG20 or we have progesterone. Okay. So, these are two hormone containing, hormone releasing intrauterine devices. Lips loop also doesn't contain any hormones, so option D is also incorrect one. The contraceptive Saheli. This is a very important question. We have question number 31. We have Saheli. What is Saheli? It is the world's first and only oral non-steroidal contraceptive pill. It is an oral pill and it is non-steroidal. And usually in oral pills, we have to take one pill every single day. But Saheli is what? It is once a week pill. Okay. So what is its functioning based upon? It regulates or it modulates the estrogen receptor and it prevents implantation. And Saheli, a very proud moment, it is developed in India only. It is developed in India. So, what are the salient features about Saheli? It is a contraceptive pill, which is oral, non steroidal, and we have to take it once a week. What are its function? How does it act? It prevents implantation and it also modulates estrogen receptor, which is our. So it blocks estrogen receptors in the uterus and it prevents it prevents eggs from getting implanted. Next, we have the function of copper ions in releasing intrauterine devices. Question number 13. We have copper releasing IUDs. Example is copper T or multiload 375. What are the functions of copper? First of all is that it decreases sperm motility. That fertilization doesn't take place. Second is that it increases phagocytosis of sperms, which also results in no fertilization taking place. In case by any chance fertilization takes place, it inhibits implantation. Okay. So which one is the correct one? We have option D that the copper ions they suppress sperm motility and they decrease the sperm fertilizing capacity of the sperms. Okay. So copper ions they interfere in the sperm movement, which suppresses the sperm motility and fertilizing capacity of sperms. The hormones present in the intrauterine devices. Um, what is the function of hormones in IUDs? They make the so of the IUDs, intrauterine devices. It can be of two types. It can be either hormonal or the other one is copper releasing. We have talked about the copper releasing ones here. Hormone releasing, they have hormones that can be progesterone mainly, or in some cases, estrogen can be present. What do? They do? They make the cervical mucus thick. They thicken the cervical mucus, and this mucus it blocks the sperm to fertilize the eggs, whereas it also prevents ovulation. Okay. So these are the mechanisms by which are hormone containing intrauterine devices work. Moving on to next, we have amniocentesis again. Question number 33. Amniocentesis. Amniocentesis is usually done when amniocentesis, which is known as amniotic fluid test, is a medical procedure which is done by it is done for either prenatal. Prenatal means what? Before a pregnancy, before parturition, before delivery, we are doing it for prenatal diagnosis of chromosomal abnormality and fetal infections. It is also done for sex determination, and due to female feticide, this has been banned in India and it is illegal. Okay. So, how is this process done? In this, we take a small amount of amniotic fluid, which contains fetal tissues, and it can be done for the diagnosis of cleft palate. So, what is cleft palate? It is a developmental abnormality which can only be detected by sonography. Okay. So, it is done between 16 to 20 weeks of pregnancy. What all can be screened with the help of this test? We can screen abnormalities such as Down syndrome. We can diagnose cystic fibrosis or spina bifida. Okay. Now, next question we have: which of the following approaches does not give the defined action of contraceptive? Okay. So it is a match the following. We have to match the contraceptive with their action. First is barrier method. So barrier method, it acts as an obstruction, so it does not allow any. This one is question number 34. So barrier method prevents fertilization, which is correct. Then we have intrauterine devices. These intrauterine devices can be of two types. It can be hormone containing or it can be copper containing. So copper containing ones, they increase the phagocytosis of sperm. They suppress the sperm motility and they decrease the fertilizing capacity of sperms. Okay. Next, we have hormonal contraceptives. What is the function of hormonal contraceptive? They prevent or retard the entry of sperm. How do they retard the entry of sperm? They actually make the cervical mucus thick. So, hormonal contraceptives make the cervical mucus thick. And what is the benefit? It increases the phagocytosis. It increases the phagocytosis of sperms. This is actually for copper containing. By making the cervical mucus thick, they are preventing the entry of sperms. Okay. And also they prevent ovulation and fertilization. Vasectomy is what? We are ligating the vas deferens. So spermatogenesis is actually taking place. So spermatogenesis is taking place. The point is there is no release of sperms into the semen. So the semen, it actually doesn't contain any sperm. Semen does not contain any sperm. Okay. So the option D is actually the incorrect one. There is spermatogenesis, but the semen doesn't contain that sperm. Okay, so vasectomy, it is a surgical procedure for male sterilization, and it is a permanent method of contraception. What do we do this during this procedure? The male vas deferens are severed and then tied in a manner to prevent sperm from entering into the seminal stream. Okay, so hysterectomy, we have question number 35. Hysterectomy is a process in which we remove the uterus. So, hysterectomy, in hysterectomy, uterus itself is removed from the human body. And why is hysterectomy done? It is a very, like, we do hysterectomy only in terminal, like, we do not have any other option left. It can be done either after menopause. And why do we do it after menopause? If there is uterine fibroids, there are fibroids present in the wall of uterus, or there is a cancerous growth. If we have uterine cancer, we'll remove out the cancer, we'll remove out the uterus as a whole. So, what is uterine fibroid? Uterine fibroid are actually benign. They are non-cancerous growth in the uterus that can develop during a woman's childbearing years. So these fibroids, they abnormal bleeding and pain in the women. So these are the characteristics of a woman. These are the symptoms that are seen in a woman who is having a fibroid. So fibroid, they are generally detected by USG. We do an ultrasound. Ultrasound is done in which we can detect if any fibroids are present in the uterus, in the uterine wall of that particular female. And in case we find that fibroids are present, we remove the uterus as a whole so that it doesn't cause any further symptoms or pain to the patient. Okay. So this was the last question. With this, we complete our chapter reproductive health. We have done all the previous year questions. Thank you.